Hi friends, good morning to all. In this video lecture, we're going to study microbiology of meat, poultry as well as seafoods. In previous lectures, we have seen microbial spoilage of fruit juices, then vegetables, then bakery products. In this video lecture, we're going to study microbial spoilage of meat as well as poultry and seafoods. As far as the meat, poultry, and seafoods are concerned, so they are of living origin and they are originated from some living sources and they contain lots of nutrient and besides water. So these are highly perishable foods as far as their classification is concerned. So the microbiological profile of meat product presented to consumers is the sum of total of slaughtered animal health and the condition under which it was reared. So basically the microbiological profile is associated with these two conditions, that is slaughtered animal, the health of animal which is going to be slaughtered, and the conditions under which it was reared. So both these factors, they affect microbiology of meat as well as poultry product, and the quality of slaughtering, processing, packaging and conditions under which the meat was stored. So all these factors, they are responsible for microbial load, and meat pathogens can cause self-limiting human enteric diseases and systemic fatal infections infections of immunocompromised and elderly and young. So meat can act as an ideal substrate for microbial proliferation as it contains all essential nutrient, and major meat associated pathogenic bacteria include Clostridium perfringens, then Staphylococcus aureus, then Salmonella species, and pathogenic strains of Escherichia coli, then Campylobacter species, Yersinia enterocolitica, then Listeria monocytogenes, and Aeromonas hydrophilia. So all these are pathogens, they are associated with meat, and microorganism associated with meat during processing includes, so meat spoilage indicates color change. So these are the features. By diagnosing these features, we can identify spoilage of meat. So the feature includes color change, then textural change, and development of flavors and of odor or slime as a result of microbial growth. So Salmonella is the primary microbial challenge for poultry, and the primary microbial to beef industry is Escherichia coli. Then Listeria, which is an adulterant with zero tolerance, is the major problem to ready to eat meat products, and treatment with organic acids, hot water steam, then pasteurization and steam carcass vacuuming, then trisodium phosphate, acidified sodium chloride, then chloride dioxide, then lactoferrins, then peroxylic acid, then sodium lactate, then sodium acetate, sodium diacetate, ozone radiation have been used as a microbial decontaminants during the meat processing operations. So these are some chemical and physical factors that are used to remove microbial load or pathogens from meat as well as poultry product. So carcass washing with hot water up to 80 degrees Celsius for 10 seconds can reduce microbial load by 2 logs, and current regulatory policies and inspection in meat industry includes HACCP, that is Hazard Analysis Critical Control Point, food safety system with an objective to provide a safe food for consumption and prevent chemical, physical as well as biological hazard. So as far as gram-negative bacteria are concerned, so these are basically aerobes, and it contains Neisseria, then Psychobacter immobilis, then Acinetobacter, then Acinetobacter jonesii, then Pseudomonas, genera includes Pseudomonas fluorescens, Pseudomonas fragi, Pseudomonas putida, and in case of gram-positive bacteria, it includes Staphylococcus species, then Clostridium species, then majority Clostridium species, they are prevalent as far as gram-negative bacteria are concerned, and all these are concerned with microbial spoilage of meat as well as poultry product. So in case of fresh meat, so it is spoiled by different kinds of microorganism, and in contrast to fruits and vegetables, meats are composed mainly of proteins and fats rather than carbohydrate. So these are protein and fat rich commodities. So water content is up to 71 to 76%, and therefore moisture is not an issue except for spoilage of microbes on curd meat. So these are highly moisture containing foods, and they contain lots of water. So due to this high water activity, these are highly perishable commodities due to microbial spoilage. So mussels, sorry for the interruption. uh, Mussels and healthy animals do not contain any bacteria or fungi, but as soon as the animal they get slaughtered, meat is exposed to contaminants, and good sanitization practices are essential to produce high quality meat. So intact animal it is less susceptible for microbial spoilage, but after that animal it gets slaughtered, it is all tissues and muscles they get exposed, and they are susceptible for microbial spoilage. So hygienic condition as well as good sanitization practices are essential to produce high quality meat. A number of spoilage organism on meat just after slaughter is critical factor in determining its shelf life. So the surface of bees crasses may contain anywhere from 10 raised to 1 to 10 raised to 7 CFU per centimeter square, and most of which are cyclotropic bacteria. Chopping and grinding of meat can increase microbial load as more surface area is exposed and more water and nutrients become available. So a large variety of microbes are commonly found on fresh meat, but different microbes become dominant during spoilage of different meat depending upon pH, then composition, then texture of processed meat, then temperature and packaging and atmosphere. So Pseudomonas species is the most predominant spoilage bacteria in aerobically stored raw meat as well as poultry. Once the initial low levels of glucose are depleted by various microbes, so Pseudomonas has an advantage because it can oxidize gluconates and amino acids more readily than other microbes, and breakdown of these compounds result in production of malodorous sulfide, then ammonia and amines including biogenic amines, then other products, and dark firm and dry meat with a relatively high pH that is up to 6.0 spoils more rapidly because deamination of amino acids start earlier. Salmonella putrefactions does not grow on meat below pH greater the pH above, which is maintained above than 6, and but can produce sulfides and ammonia even when glucose is still available. So these sulfides not only smell bad but also cause color change in meat, and therefore salmonella has high spoilage potential on fresh high pH meats stored aerobically even when it is not dominant microbe. Then Brocothrix thermospatica is often significant spoilage organism on fresh meat stored aerobically at refrigeration temperature, and Enterobacter, particularly species of Serratia, then Enterobacter and Hafnia are major cause of spoilage in vacuum packed high pH fresh meat. So these organisms are facultative anaerobe that produces organic acids, hydrogen sulfide, and greening of meat. So lactic acid bacteria grow on meat and poultry packed under vacuum and modified atmosphere, producing organic acids from glucose by fermentation, and these gives rise to acidic of flavors which may be accompanied by gas as well as slime formation and greening of meat. However, lactic acid bacteria are weakly proteolytic and so do not produce large amount of amines and sulfides, and spoilage of meat by lactic acid bacteria is not offensive. So saccharophilic and anaerobic Clostridium species are associated with spoilage of vacuum packed meats. Then blown packed meat spoilage is characterized by excessive gas formation with off odors due to formation of butyric acids, then butanol and sulfurous compounds. So yeast and molds grow relatively slowly on fresh meat and do not compete well with bacteria. So therefore they are minor component of spoilage of flora. So overall bacteria are more predominant in case of spoilage of meat is concerned. Then we have seen this spoilage of fresh meat. So here spoilage of processed meat, how it get occur. So addition of sodium chloride, then nitrate and or nitrate along with various other seasoning, emulsifiers and preservatives go ground or whole muscle meat changes the environment significantly and also spoilage flora of processed meat. So dried and dry fermented meat generally do not support microbial growth, although process deviations may allow growth of some organisms. Spoilage organisms can grow on fresh and cooked cured meat, so they are best stored in chilled under vacuum or modified atmosphere. Pseudomonas species are not usually important cause of spoilage in processed meat because of their sensitivity to curing salts and heat pasteurization and their inability to grow well in meat packed within vacuum or high carbon dioxide atmosphere. However, when packaged have been opened and there has been insufficient curing, so these bacteria may spoil refrigerated processed meat, and some cold and salt tolerant enterobacteriaceae have been caused spoilage in some specific processed meat such as ham and bacon. So lactic acid bacteria is a group of bacteria primarily associated with spoilage of processed meat. So they produce so raw flowers, then gas, then slime and greening, and these spoilage may be more severe than fresh meat because of presence of added carbohydrates. So competitive ability of different lactic acid bacteria strains is related to pH, water activity of meat, cooking, then storage temperature and oxygen and carbon dioxide levels. So spore formers such as Clostridium bacillus are usually not spoilage problem in processed meat because of presence of nitrate and other curing salts. However, faulty cooking and cooking procedures including long cooling periods and temperature abuse has allowed growth of these organisms in some cases. So spores of these organisms may be introduced with species other than ingredients. So yeast cause some spoilage in processed meat but are generally only important when sulfite is used as a preservative or when meat have been eradicated or are stored aerobically in the cold. So slime may be produced along with visionary or malty of flavors in some sausage. So these are spoilage of processed meat. So here spoilage under aerobic condition. So it's surface slime caused by Pseudomonas acinetobacter, then Moraxella alkaligenes, then Streptococcus, then Leuconostoc and Bacillus micrococcus as well. Then change in color of meat pigment, it is due to red color of meat maybe change to shades of green, brown or grey by lactobacillides Leuconostoc species. Then changes in fat content, they are induced by some lipolactic bacteria which produces off odors due to hydrolysis of fats and production of aldehydes as well as acids, and these type of spoilage is caused by lipolactic Pseudomonas as well as Chromobacter and yeast. Then surface color change, it may be occurred due to red pigment producing bacteria Serratia, then it causes a red spot on meat. Then blue color surface is caused by Pseudomonas, then yellow color is caused by Micrococcus species. Then off odors in case of spoiled meat and off taste is caused by different kinds of yeast as well as Actinomyces species, and volatile acid like formic acid, acetic acid, butyric acid as well as propionic acid produce sour odor, and Actinomyces produce musty earthy flavor. So yeast also cause sliminess, discoloration and off odors and taste infects. So in case of aerobic molds also cause spoilage of meat. So these are stickiness, viscous, black spot, white spot, green patches, and off odors of taste. Then spoilage under anaerobic condition includes soaring, and it is caused by production of formic acid, then acetic acid, then butyric acid, then lactic acid, and succinic and propionic acids. Then putrefaction is caused under anaerobic condition, and it is caused by decomposition of proteins and under anaerobic condition by Clostridium species, and foul smell is due to production of hydrogen sulfide as well as mercaptans, then indole and scatol and ammonia and amines. So these are photographs and photos. You can see spoiled meat. It is processed as well as raw meat you can see, and these are different kinds of spoilage. In case of eggs, so freshly laid eggs are generally sterile, particularly inner content. However, shells get contaminated from environmental sources such as fecal matter of bird, then beddings by the handlers, and wash water. Also, the packaging material in which eggs are packed. So these are factors that affects spoilage of egg, and they are main responsible sources of spoilage of egg. So there are several extrinsic and intrinsic mechanisms through which eggs are protected from microbial invasion. So waxy shell membrane retards entry of microorganism. Then further shell also prevents entry of microorganism, and membrane inside shell behaves as mechanical barriers to entry of various kinds of pathogens as well as microorganism. And further, lysozyme present in egg white is effective gram-positive bacteria is effective in eliminating against gram-positive bacteria, and avidin in egg white forms complex with biotin, thus make it unavailable for microorganism. So also high pH of egg, that is up to pH 9.9 to 10 of albumin, inhibits microbial growth. Then binding of riboflavin by apoprotein, and chelatin of iron, chelation of iron by cannibulation further helps in hindering of growth of microorganism might be gained entry inside egg. So this is structure of egg. So by default, chemical compound present in egg and some physical as well as mechanical barriers, they prevent entry of microorganism as well as pathogens. So this is privilege. So spoilage of egg occurs due to species of Pseudomonas, then Acinetobacter and other species, and breaks or cracks in egg shells taking place due to transportation or mechanical damage may allow microorganism to enter into the eggs. Otherwise, intact membranes and intact shells, they resist entry of pathogens as well as different kinds of microorganism. So once they entered into yolk sac, causes spoilage and storage. So eggs on storage may lose moisture and therefore weight. So white of egg becomes thinner and more watery on storage. The major changes in egg takes place during spoilage. Organism in general, spoilage of eggs is caused by bacteria composed of molds and can be described as a green rot, due to which growth of Pseudomonas fluorescens, then colorless rot due to growth of Pseudomonas, then Acinetobacter and other species, and black rot due to Proteus and Pseudomonas, red rot due to Serratia species, and crossed rot due to Proteus vulgaris and Pseudomonas intermedium. So growth of Aeromonas in yolk sac turns in black color, and also there is a strong putrid odor due to formation of hydrogen sulfide, and storage of eggs in high humid atmosphere may help in growth of several modes on surface of egg shells. So mode causing spoilage of eggs includes species of Penicillium, then Mucor and Alternaria, etc. Then in case of poultry meat, so poultry meat like meat and other animals also susceptible to contamination by various sources. So contamination of skin and lining of body cavity takes place during various process operation, and the organisms are of great importance in poultry are Salmonella species, then Campylobacter jejuni and several gram-negative cyclotropic bacteria, visually Pseudomonas, Acinetobacter and Flavobacterium have also been isolated from poultry carcasses. So ground turkey also may be carry fecal streptococci. It is important to freeze the poultry fast in order to keep it in good condition for several months. So freezing further reduces the number of microorganisms in poultry meat, provided with temperature is maintained quite low at below 18 degrees Celsius or below. And in case of fish spoilage, so fish is a very perishable commodity, and due to high protein food that typically contain high level of free amino acids, and the lipid content of fish is up to 25%, and it has very low content of connective tissue, that is approximately 3% of total weight, as compared to with not around 15% in meat. So fish flesh generally contains 15 to 20% of protein and less than 1% of carbohydrate. So non-fatty fish such as cod's haddock whitening fat levels are only about 0.5%, while in fatty fish such as mackerel, herring levels can be vary between 3 to 25%. So overall composition of fish, you can see that water it contains 60 to 80% of water, then fat it extend up to 1 to 20%, and protein they are up to 14 to 20%. So microbes metabolize these amino acids producing ammonia, then biogenic amines such as putrescine, then histamine and cadaverine, then organic acids such as ketone and sulfur compounds, and degradation of lipids in fatty fish produces rancid odors. And in addition, marine fish and some freshwater fish contains trimethylene oxide that is degraded by several spoilage bacteria to trimethylamine, that is TMA, and the compound responsible for fishy of odors. Iron is a limiting nutrient in fish, and this favors the growth of bacteria such as Pseudomonas that produces siderophore that bind iron. So these are chelating agents, and spoilage bacteria differ somewhat from freshwater and marine fish for temperate and tropical water fish. So storage and processing condition also affect microbial growth. So Pseudomonas and Savanilla are the predominant species on chilled fresh fish under aerobic condition, while packing under carbon dioxide and addition of low concentration of sodium chloride favor growth of lactic acid bacteria and Photobacterium Phosphorium. So heavily wet salted fish supports growth of yeast, while dried and salted fish are spoiled by molds. So addition of organic acid select for lactic acid bacteria and yeast. Pasteurization kills vegetative bacteria, but spores of Clostridium and Bacillus they still survive, and they may grow particularly in unsalted fish. So spoilage of fish and seafoods, this is mainly caused by halophilic bacteria like Serratia, then Micrococcus, then Bacillus alkaligenes, and Pseudomonas causes spoilage of salt fish, as well as shellfish are spoiled by Acinetobacter and Moraxella and Vibrio species. Then crab meat is spoiled by Pseudomonas, Acinetobacter and Moraxella at low temperature and by Proteus at high temperature. So microbial load in shrimps, oysters and clams depend on quality of water from which they are harvested. If the sewage is drained to water bodies, microbial quality deteriorates. So during handling, fecal coliforms, fecal streptococci and Staphylococcus aureus may be incorporated into this product, and salmonella also is found in oysters, possibly due to contaminated water. So seafood also is the source of Pseudomonas species, then Clostridium perfringens and Listeria monocytogenes, then Vibrio parahemolyticus, as well as Salmonella enteritica, servor, then Enteritis and Typhimurium, then Campylobacter jejuni, then Yersinia enterocolitica and enterovirus, that is hepatitis A. So smoked salmon and shrimps also found to carry pathogens like Listeria monocytogenes. So these are overall scenario for microbial, which type of microbes they causes different kinds of fish as well as seafoods. Then there are several meat-borne diseases. So foodborne microbial hazards have devastating impact on human suffering. So microbial pathogens are of current concern that need to be controlled in fresh meat. Includes Salmonella, Campylobacter, enteropathogenic, Escherichia coli, includes serotype and enteric pathogen other enteric pathogens. Infection due to Listeria monocytogen following consumption of ready-to-eat meat as well as poultry product is a major problem in recent years. Also, there are foodborne infections caused by Yersinia enterocolitica, then Staphylococcus aureus, and Clostridium botulinum, then Clostridium perfringens, and Bacillus cereus. So, prevalence of some of the foodborne pathogens recognized since 1970 includes Vibrio cholerae, then Vibrio maleficus, then norovirus, and Enterobacter sakalazi. So, prions and resistant bacteria, so these are of pretty much concern. And in recent years, the foodborne pathogens associated with animal health pandemics includes avian influenza, then foot and mouth diseases, viruses, and avian influenza is not of concern to poultry meat's safety because it is inactivated by proper cooking with temperature at 70 degrees Celsius and more. Also, oral route of transmission is less important than other non-foodborne route. So, presently there is a continuous adaptation and development of resistance by these pathogenic microorganisms to antibiotics and potentially to traditional food preservation barriers like low pH, heat, cold treatments, then dryness, then low water activity and chemical additives. So development of antibiotic resistance in foodborne pathogen is very important as far as public health point of view is concerned, and the situation gets problematic day by day. So as far as control of these meat-borne pathogens. So effective control of meat-borne pathogens and enhancement of meat safety can be achieved by control of latent infections among livestock, then animal welfare and human treatment, as well as application of antimicrobial interventions at the farm during harvesting, dressing and product processing. Then improvement in processed food hygiene and potential application of new and novel processing and preservation technologies, and animal stress can damage meat quality and lead to more contamination and increased pathogen shedding. Then antimicrobial inversion technologies can be used effectively to improve the microbiological quality of meat, and these technologies include reduction of contamination of raw product, then minimization of microbial access to product, reduction of contamination that has gained access to product, inactivation of microbes on a product without cross contamination, and prevention and control of growth of non-activated microbes which have gained access to milk. So an effective pathogen control at pre-harvest, post-harvest, processing, storage, distribution, then merchandising, preparation, food service and consumption of meat includes activities employed during post-harvest or in field. So during the post-harvest or processing in plants, retail food service and home pre-harvest pathogen control invention includes diet manipulation, then use of food additives, then antibiotics, bacteriophages therapy and immunization of these animals. So overall these are a process of prevention of meat-borne infections, and these are some standard processes that is HACCP system, then GMP practices and GMP, then antimicrobial, all these substances that can use for prevention of microbial control of meat-borne pathogens or foodborne infection that are caused due to meat as well as poultry as well as sea products. So thanks for this lecture. We'll see another topic in next lecture. Thank you.